And the remarkable thing was that actually that type of change and what we're now seeing in growth in children during the first 4 months of life show that these proinsulin levels, are different by sometimes as much as 32%. Now, is 32%, we heard Ronnie talking about percentages, whether they're big or not. Well, are there differences in 32% important, as far as physiology is concerned? Well, the internists say that a difference in normal above 35%, okay, defines a pre-diabetic state. So the kinds of things that can be done in the first 4 weeks or the first 4 months of life actually can program you metabolically for some rather significant problems. 20% of the risk of obesity, that is a BMI greater than 95th percentile in adults, can be attributed to the rate of weight gain in the first 4 months of life. So my conclusions are that pediatricians will use a lot of different things, and you've heard about them today. We'll use the genetic code, and that'll help us with about 20 to 30% of the variation. We'll use gene expression. You saw lots of pictures of gene expression. We'll actually use the products of the genes. We'll get away from the genome and actually look at proteins. They're actually more accessible and actually giving us more information. And that will help us predict susceptibility to common complex adult and onset diseases long before they become symptomatic. But my prediction is, and I've shown you ways how we have harmed or we have altered the program and how we've reversed the program, is that pediatricians will also be able to reprogram both prenatal and postnatal genetic programs to alter the risk factors of complex, common, and adult onset diseases. Now I'm going to close with being interested in sickle cell disease. I always like to show you this. One of the oldest ideas about evolution and how it affects humans comes from the study of sickle cell disease. Actually, the mutation for sickle cell disease probably occurred independently in these 3 areas of Africa and also somewhere between India and the east coast of Saudi Arabia. And because those areas were also endemic for malaria, it turns out that if you have a sickle trait, okay, rather than AA hemoglobin, we actually know now that that protein sitting inside the red cell stimulates a certain enzyme. And that enzyme actually inhibits the growth of the malarial parasite. So that what happened by selection was sickle trait was favored, okay, more than AA, the normal individual. Unfortunately, the ones who got the double dose of S, sickle cell patients, actually were selected against. So a sickle trait can be as high as 25 to 30% in some areas of Africa and in areas of India. So here's a way where the environment selected for postnatally a gene to become more frequent.
Is that a recessive?
Hmm?
Is it a recessive?
If you would call it recessive because AS supposedly has an innocent phenotype. Now, unfortunately, I've gotten involved in a very big controversy. You may have read about this, about AS individuals. Those are individuals with sickle trait and whether they should be screened. The NCAA, not because of any science and completely ignoring everything we know about genetic screening, has now decided to screen all sickle trait athletes only in the AAA or the top group of athletes. So if you happen not to get into the best schools, but you have to play football in the lower schools, they don't have to screen you, okay? But it's the most inane thing that has to do with genetic screening that's ever happened that in my 40 years of seeing this. But sickle trait may turn out to have some physiologic significance. So I'm not certain how to define autosomal dominant and recessive on the basis of phenotype. But most of us say it's a recessive. So here's what I think's going on is, let's say there's an adverse prenatal environment. Let's say that there's just not enough kilocalories going to the baby or to the mother. Well, what we know then is that there will be genes that we now identify with being associated with obesity, type 2 diabetes, and high blood pressure. Now, those genes aren't there to do that. Those genes are there to do lots of other things. We just associate them with those diseases because we don't know yet what the physiology is. But we'll call them those genes just because they're there. Well, those genes actually get either turned up or turned down. And as a result of that, you see a low birth weight infant. Now, there's physiology that would say that actually when these genes are in this way, that if that infant then enters an environment where there's very few kilocalories, their metabolism will be slower, okay? And actually this will help them survive if in fact they're going to enter an environment somewhat like what they were, the environment in utero. But if there is a, quote, adverse postnatal environment, either physicians trying to make babies grow fast or parents trying to push babies up growth curves, okay? I have another talk on the tyranny of the growth curve, in which, you know, all of us have raised our children wanting them to be above the 50th percentile. And there was probably a reason that there was something below the 50th percentile. But if that happens, then in fact you predispose in later life these infants to obesity, type 2 diabetes, increased blood pressure. And in fact, these things are quite interrelated, these disorders. Okay? So what I think will happen is, one, pediatricians will become the reprogrammers. Not by looking at birth weight, but looking at the biology of how these genes control the weight and what's different and what are the detections. I think that the pediatricians are going to have a whole new repertoire of things that they can do to prevent adult diseases. And I think we can begin to monitor these reprogramming events prenatally. And some of the phenomenal new technology we have now says that not only can we can monitor the DNA in an unborn baby, we can also monitor the RNA expression in unborn infants just by taking blood from the mother. So that this is becoming a very, very accessible way to begin to sort of sort out what this is all about. In summary then, I think the environment's very important, and I will suggest to you that when all is said and done, the Genome-Wide Association people are going to find enough SNPs to explain about 20 to 30, maybe 40% of the risk for most common disorders. No more. And I don't care how fancy they get with copy number variation and all these other things, they're not going to be more than that. Okay? The real thing is that the whole other half of the story is how the genes get turned on and off subject to the environment. Okay? And so the environment actually manipulates the program, and I've used the light bulb here to say it. But pediatricians are going to be able, by understanding those programs, actually to reverse some of those issues. Thank you very much. How many times has this book shown up today?
I quoted them in the first
Did you
slide.
show the picture?
I did show the picture.
Okay. This is all of you who are serious about science and genetics, you better read this book. This book is by Barton Childs. It's called Genetic Medicine, A Logic of Disease. And this book actually outlines the curriculum for the Hopkins medical students. But it has two very important ways that it actually explains what's going to happen to medicine in the next 50 years. One, we're going to treat individuals and not diseases. And two, that health is defined as a homeostatic mechanism between the genes and the environment. And if you understand those two fundamental principles, and he lays this all out for you, then you're going to understand all the major, really, things that are going to happen in genomics and in medicine for the next 25 to 30 years. Thank you.